Good afternoon, everyone, and welcome to today's webinar on Western Blotting and Stain-Free Technology, hosted by Lasec in collaboration with Bio-Rad. We are pleased to have Arda Akalin with us today as our speaker. Arda has more than 11 years of experience in life science, commercial and application experience, and is currently working for Bio-Rad as a business development manager, responsible for regions such as Turkey and Sub-Saharan Africa, mainly focusing on the Proteomics division. Before I hand over to Arda, kindly note that we've disabled the chat function. However, I would like to encourage you to participate using the Q&A feature, which can be found at the top of your screen. So without further ado, please join me in welcoming Arda. Thanks, Arda. Over to you.
Thanks, Michelle. Hello, everybody. I'm really appreciate to have a meeting with you, and I really appreciate to explain our solutions related to Western blotting. And as Michelle mentioned, there is a question and answer section and also chat window. If you would like to ask any question, please don't hesitate about that. And I will also ask several questions to you just for fun, and I really appreciate if you respond. As Michelle mentioned, I'm working in Bio-Rad for long years, and I am responsible related to Western blotting, protein purification chromatography, and multiplex immunoassay. Also related to other topics, you can directly contact to us for further information. Today, I'm going to talk about mainly for stain-free Western blotting and total protein normalization, because you will see that why the normalization is really important, and is it still time consuming to run your Western blot experiment? And also there are several advantages and disadvantages using housekeeping proteins, as well as I'm going to explain how we can get really quality results using this stain-free Western workflow. And we have a new imaging system. I would like to give a brief information about this new chemiluminescence ChemiDoc Go system. And I would like to share a real customer example, how you can easily validate your Western blot data using housekeeping proteins and stain-free total protein normalization. Before we start, I would like to thank Lasec team, because we work in these EMEA channel partner regions with our partners. So Lasec is our official channel partner related to sales, service, applications, and training. So I would like to thank them again to prepare these nice atmospheres with you and to have this presentation. And I have a quick question before we start. Who is the better high jumper? And respond, I really appreciate. Is it human being? Is it grasshopper? Or is it dog flea? Which one do you think can jump higher or better high jumper?
Hi, Arda. Let me give it a crack, seeing that everyone else would have to answer through the Q&A section. My guess would be it's really dependent on what you're looking at in terms of what variables you're looking at. So is it body to mass ratio? Is it the height?
Yeah, actually, it's something good that you ask these questions. You know, we need to define if we are talking about the higher jump, it should be according to what? Like it's also something like normalization, because in case we think about that, which one is higher jumper, and when we normalize this data according to their body weight, body length, so we may identify that even the shortest distance is 25 centimeter, but when we normalize this data according to this dog flea's size, it can jump 200 times more than its body height, body length. So normalization is really important when we talk about scientific data which will be published. So according to, there are several highly important papers which talks about normalization and signal range. So for quantitative comparison of anything that you are going to publish, it should be in the linear signal range, and it's highly important for any type of experiment. And this information comes from Nature. And the others, which comes from Journal of Biological Chemistry. And when you have any data, we need to confirm that according to antigen loading or normalization, it should be in a regular linear signal intensity. It shouldn't be saturated. And if possible, we need to normalize the signal intensity according to total protein. Whenever possible, it's also a recommendation. And, you know, as most of us experienced about the housekeeping proteins instability related to normalization, it's recommended or requested. You should able to prove that manipulation doesn't affect your expression level on the housekeeping protein. I will also give several examples on the next slides. And as you know, accurate normalization requires that your signals should be in linear range and it shouldn't be saturated, as the example on this publication. And as you see, some of the housekeeping protein signals are saturated or differs from one to the other. And it should be detectable when differences in the signal intensity related to your samples or related to your antibody amount, incubation time, and you should be able to detect this variation also in your blood. And of course, the loading control must be unaffected from your experimental conditions. And when you run your experiment and normalize your data according to your expression level of loading control, and if there is any problem, so it will result with misleading results of your experiment. So how we can help? We have a technical note related to how we can use total protein normalization to get better results from your, or reliable results from your Western blotting experiment. We will be able to share this document to you by email, but I would like to touch another point, how long time we do spend running this Western blot, because as everybody has experience, it takes quite a long time comparing other biological or molecular biology related tools, because preparation takes quite long, running a gel takes quite long, then antibody incubations takes quite long, and optimizing all the different parameters takes quite long. Instead, like comparing a qPCR experiment which can be completed in 2-3 hours, or multiplex ELISA, even with hands-on time, it can be completed in 3-4 hours. The Western blot, traditional Western blot, may take quite longer than the other molecular biology tools. But how we can help related to using our precast or fast-cast solutions means that we do provide precast gels, yes, but we do also provide fast-cast gel solution kit that you can cast your own stain-free gel, and you can use these gels as precast quality gel, shorter running time, really short preparation time, compatible with stain-free and quick blotting. So I will give some time-related information right now. As you know, when you cast your own gel, preparation and polymerization takes approximately like more than 1 hour, and you need to run your gel first like lower voltages, then you need to increase voltage, and it takes approximately 1 additional hour. And in addition to this, we need to transfer our gels to the membrane at least 1.5 hour, even in some cases overnight transfer using wet transfer. So in as total, in best scenario, it will take approximately 300 minutes. But in our fast cast solutions, that you will be able to prepare your own gel using our solutions, and it takes just 5 minutes because everything is ready in a bottles. I will show details related to this. Then you will be able to run these gels in 300 volts. I mentioned here like 250 volts, which is similar to our precast gels. Accordingly, your gel will be run completely. And on the other hand, there is another blotting pack preparation which is cost effective, and we are preparing this ready to assemble transfer packs, and these packs can be used using Transplot Turbo for quick transfer. Transplot Turbo works from 3 to 10 minutes. So according to I mentioned here the longest time, which is 10 minutes. When you cast your gel, run your gel, then you prepare your own transfer pack using this ready to assemble kit. Then when you transfer your gels to the membrane, it takes just 75 minutes. Comparing this stain-free precast gels, it's approximately 35 minutes. So it's, of course, these two technology helps to save a lot of time, but not it's like that. In other cases, you need to run, of course, your primary antibodies, secondary antibodies. There are wash steps. For this example, I accept your primary antibodies incubation time just 1 hour in room temperature for all these different cases. But when I need to run my housekeeping protein for housekeeping protein normalization, I need to consider maybe reprobing, like stripping and reprobing and secondary antibody incubations, which doubles my expenses, also my time. So according to when I spend all this time to complete the housekeeping protein normalized traditional Western blot, I need to spend approximately 10 hours. Of course, it may differ, but according to this condition, it's 10 hours minimum. But in case I run stain-free precast gels or fast cast solution kit, I can reduce this time down to 3.9 hours, maximum, let's say 5 hours, from 3 to 4 hours. And as you see here, in case I run total protein normalization instead of housekeeping protein normalization, I can really save a lot of time plus reagent. So I will explain how it will be possible. So in stain-free Western workflow, as a technologically, we can detect proteins on the gel from 0.2 to 5 nanogram, which is compatible or the same sensitivity comparing silver or comesa staining. And we do provide really good and wide linear dynamic range. Accordingly, in this example, as you see, when we have serial dilation of high protein, wide dynamic range of proteins in the gel from 80 microgram to 2.5 microgram. And when we take the all lane protein information to the graph, and when we see that the dynamic range detection range is quite linear, and which called higher dynamic range, but also when we have a serial dilation of the lysate from 20 to 1 microgram on the gel, and when we get the total lane intensity for each lane as a graph, so we will be able to detect really good accuracy, which is 0.984, and here as you see 0.999. So this technology works quite accurate. On the other hand, the signal intensity of the stain-free gels are much higher and stronger than the, let's say, Ponceau stain membranes, and also they provide superior linear dynamic range. Plus, this technology is more effective. It's specifically important in case small fold differences in your protein expression is highly important. They provide quite better results, more effective results compared to housekeeping proteins. And the other important point, when you run stripping applications, there are several important considerations related to primary antibodies that you need to confirm you completely remove primary antibody from the first application. So this step mostly skipped because it requires re-incubation of the blood with ECL and taking exposure on the film or imaging system, and you need to spend ECL and antibody again, and then you need to reprobe and you need to control with stripping. So when we skip this step, we may fail because of the remaining primary antibodies from the first blotting experiment. So it may bring problems as well. So because the stain-free technology is not required immunodetection of housekeeping proteins, accordingly, reprobing steps can be, of course, avoided altogether. So you don't need to run these steps. And in addition, when you think about housekeeping protein, in some cases, because they are already expressed too much, comparing your low abundant protein, you may get oversaturated bands, or you may have different expression levels on the proteins. And so accordingly, when you run your experiment and normalize your housekeeping protein normalization, you may prone to errors of the data interferences. And on the other hand, housekeeping proteins are subject to change according to different tissues and also under different disease or pathological states and under different various of experimental conditions and during various developmental stage or different genetic backgrounds. So in case you are using a housekeeping protein normalization, you need to validate your housekeeping proteins never change according to these different conditions, and you need to validate it. So accordingly, you can easily use your housekeeping proteins for normalization. So how this stain-free Western workflow helps you instead of skipping these all important steps and getting high quality results. So it's related to our gels. As I mentioned, it can be precast gel, or you can cast your own stain-free gel using fast cast solutions, which you will be able to save time. And you can use any Bio-Rad chemiluminescence imager or fluorescence imager. As you know, we have ChemiDoc and ChemiDoc MP imagers, but we recently launched this new smart and compact chemiluminescence and multiplex fluorescence imager specifically for academia and biotech research. And I will give a short introduction related to this. And it's also important using ImageLab software to run this total protein normalization. And of course, we do provide the other solutions like a quick transfer device, which is Transport Turbo, and electrophoresis systems. So, you can run any steam-free precast gel or fast-cast gel solutions. As I mentioned, fast-cast gel solutions are provided in ready-to-use format that you don't need to prepare anything. There is no need to prepare buffer pHs, concentration, just you need to cast these solutions to your glass plate, and it will get polymerized in 30 minutes. The advantage, you don't need to wait between resolver and staker. As soon as you cast resolver, without any waiting and without any using the isopropyl alcohol or water, you can cast staker, and you can just wait 30 minutes to polymerize. So how this technology works, in our gels, we do have a specific 3 hollow compounds which interact with your proteins under the imagers, Bio-Rad imagers. So when you run your gels and when you take these gels into the imager, there is an activation of specific wavelength UV, and accordingly you will be able to see your bands like sensitive as Comasi or Comasi stained gels. Then you will be able to transfer the same gel to the membrane. So between these stages there is no staining. Then when you transfer this gel to the membrane, you can take the membrane to the imager and you will be able to visualize total proteins on the membrane. So mostly we are going to use this total protein amount on the membrane for normalization. So in this technology, we will be able to see, after run our gels, we will take the picture of the gel, and when we transfer to the membrane, we will be able to see the total protein on the membrane. And then when we run post-transfer gel on the imager, we can be sure that if there is any remaining protein or not. So it can be used as a confirmation step that your blotting successful. So it's highly important for validate your full workflow, but also it's highly important if you wanna, let's say, optimize your first stages of transfer. It will guide you that you have good transfer, you can go the same method, or you need to optimize your blotting voltage or ampere or time. So again, there is no need for staining, and because there is no need for staining, there is no need for destaining. As you know, you can use Comasi stain or you can use other stains, but after the transfer of the gel, you will have really low amount of protein that we expect. And when you stain your gels, if you don't destain this gel properly, like let's talk about Comasi staining. Okay, you can stain your gels, but Comasi also bind to the gel instead of proteins as well, and you need to have proper destaining to see the low amount of proteins, low amount of protein on the gel. So it's not so easy to detect in case your blotting experiment works perfect or not. So how we run this total protein normalization on our imagers. So we get the stain-free image for total protein, and then we get the chemiluminescence or fluorescence image from the our blotting experiment. We just load these two images and we choose the lanes for total protein and we choose the bands for chemiluminescence. And when we integrate all these data, so it comes automatically that you will see your normalization factor and you will see your normalization band intensity as a Excel table or ImageLab table. So it's quite easy and simple, and all these procedures takes just 5 to 10 minutes. It's just related to choose the correct shape of the lanes and detecting the bands. That's all. So as you see, it's quite easy. So what we do provide is this total stain-free workflow. When we compare according to traditional Western blot, we will try to run less gel and spend less amount of sample because you don't need to repeat this experiment as much as possible. You can complete everything, almost everything in a single gel and membrane. So according to you can complete hands-on time in shorter, and you can identify any problem in advance, so you can reduce possible gel region antibody usage, or you can save time as much as possible. And the stain-free images are fluorescence. Accordingly, you will get low background, you will get a high intensity, you will skip stainings, and you will never consider if this staining is required or efficient or not. And we provide normalized results. Even your housekeeping protein fail, or you can normalize your housekeeping proteins as well, because it's total protein normalization. And we also aim reducing waste, because we will try to reduce your repeating steps in these experiments. As you know, maybe you already experienced, the most expensive experiment is, of course, repeated experiment, because we make our budgets for an experiment. But on the other hand, we also try to have some excess volume, like 10, 20, 30%. But when we need to repeat the experiment, that we double our budget in a case, so it gets the most expensive experiment in that case. I would like to give a quick introduction related to new Chemidoc Go imaging system. Then I will give examples of how we normalize total protein in our imagers. So this is a freshly new imaging system from Bio-Rad. It has latest generation of sensor. It has cloud connectivity. In case you are experienced with CFX Opus systems, which are new generation of real-time PCRs, which use Bio-Rad I/O. So these systems are also connected to Bio-Rad I/O, and it provides cloud connectivity. You will save your images directly to the server, and in case any update requires, it will update. The touch usage software will update itself automatically. So the important part is it has really small footprint. It's all LED light sources, so it's energy efficient, and you can also run DNA gels, RNA gels, colorimetric plots, chemiluminescence plots, and two-channel fluorescence plot, multiplex fluorescence, in case you need multiplexing. So it's highly comfortable, smart, and easy to use system from Bio-Rad. And lastly, I would like to give quick information about ImageLab software and normalization. So as you know, ImageLab software, really easy to use software related to detecting bands and lanes. So it's like using paint on your computer. Just when you need to detect your lanes and bands, it's really quick. Just you need to take the gel and you need to place your lanes in a really easy way. And the advantage of this software, of course, in any experiment you will have some background issues, but in this software we will be able to remove the background data from our lanes and bands. Accordingly, we will have accurate information comes from our bands, and this is real customer data which is related to expression level of a protein which is treated with medicine or drug, and the protein is MCM7. And in this example you will see the protein of interest in red channel, and you will see housekeeping protein in green channel, and you will see stain-free blot on blue channel. So as you see here, we detect lanes for stain-free blot. There is no detection of the bands because we are interested in total lane loading amount of protein, and we just detect bands on housekeeping and interest of protein. So customer decide to make normalization using our ImageLab software for as you see, housekeeping protein, which is green channel. So in ImageLab software, you need to choose housekeeping protein bands, which is green. And when you run your housekeeping protein, as you see, it accepts the first lane as a factor 1, and the second lane, it will take the intensity here as a green channel, and it will normalize it, which is the normalization factor 1.06. And when you apply this 1.06 normalization factor to your red channel, which is our interest of protein, as you see, there is no significant difference. But literature data says when you treat your sample, the MCM7 protein should change approximately 20%. But when I run this experiment according to housekeeping, it's not possible to detect. Then what we did, we take the total protein lanes for normalization. Accordingly, we choose normalization channel as blue channel, which is stain-free blot here. Let me show my pointer. Yes, here, stain-free blot, and our normalization is stain-free blot, and our normalization is lane 1. So we will normalize lane 2 according to lane 1, and we will choose, in previous experiment normalization it was housekeeping protein, but now it will be total lane protein. So when I choose this, software automatically calculates normalization factor according to total lane proteins. As you see here, total lane adjusted total lane volume, which is background excluded, is 173, and it's accepted as 1. And you will see the second lane total protein amount is 212, which is the normalization factor is 0.82. When I take this 0.82 normalization factor for my protein of interest, which is red channel, so as I mentioned, first channel is accepted as 1, and we will normalize second lane. And when I put this normalization factor, I will see is the normalized volume is 20, and here is 16. According to, I can see these differences between these two treatments, and there are almost 20% of difference. So as you see, I am not able to detect this using using housekeeping proteins, but it's quite easy to see these differences even on the same membrane using stain-free total protein normalization. So as you say, it's really quite an quick and easy approach to get your results and to normalize using stain-free images and ImageLab software, and hope it will help you to get high quality data. And I would like to appreciate if you have any question, please use question and answer or chat part, and you can send any request to us using this QR code or from our email, or you can always contact our official channel partner Lasec whenever required. I really appreciate to have a presentation with you. I really hope that we will meet again in case you need any request from our side.
Thank you again for your time and the presentation, and I hope everyone has a lovely Friday.